my friends sitting in the Zoom room with us.
Yeah.
Okay,
Okay,
cool.
I think
Yeah,
let's just get started.
cool.
Okay.
1 second. Let me share my screen.
Right. Okay, good afternoon, everyone. Welcome to the Student Sensors RIG Seminar Series. My name is Zhang Li. I'll be the chair of today's talk, and I'm also one of the members in the student-led sensors interest research group. The primary aim of our group is to share research presentations in the areas of sensors and sensing technologies. We hope the student seminar series will enable knowledge share and foster collaborations. Please visit our website to explore more about our organization. And if you would like to present your research or interested in joining our group, please do not hesitate to email us. The title of today's talk is Multimodal Optothermal Manipulation of Synthetic Particles and Biological Cells. We're very excited to have Hongru Ding here today. Hongru Ding is a PhD student in mechanical engineering, and he obtained his bachelor's degree in energy and power engineering in 2015, and his master's degrees in engineering thermal physics in 2018, both from Huazhong University of Science and Technology. His current research interest focuses on the optical manipulation of micro nanoparticles and its biological applications. Before the talk begins, I would like to remind you that you can always ask questions at any time by using the chat box. We'll collect the questions and ask the speakers at the end of the talk. Now I would like to give the floor to Hongru, and the time is yours.
Awesome. Thank you so much for the introductions, Yang. And I'm gonna share my screen. And yeah, let's get started. Good afternoon, everyone. I'm Hongru, and it's my great honor and pleasure to give this presentation. And thanks again for the invite from Sensor Rock. I obtained my PhD degree in mechanical engineering department, and specifically, I was conducting research in joint research group. Today, I will talk about multimodal optothermal manipulation of synthetic particles and biological cells. So I will mainly talk about two projects. First one, it's automated multimodal optothermal manipulation. And secondly, it's how we achieve the multimodal manipulation along various surfaces. Yeah, first, let's talk about the introduction. On this page, I will introduce the fundamental and limitations of the optical manipulation techniques. So as you can see in these schematics, it basically shows that we can use a tightly focused light or laser beam to manipulate a colloidal particle as long as the particle is very close to the focus point of the laser beam. It relies on the optical force, and so basically it's like we can use this light to twist the particle and move it around. That's why we call it optical tweezers. And it's used for the translational manipulation of tiny objects, because there are so many important applications. For example, we can use it for the measurement of cells' mechanical properties. The inventor won Nobel Prize in 2018. And based on that, some other researchers developed a light-driven rotor platform. People first use one laser beam to fix the position of the targets, and then use another laser beam to give it optical torque. That's for the rotational manipulation of tiny objects. It also shows some very important applications, for example, for the measurement of vacuum friction. We can also use this platform for the 3D cellular imaging, as you can see here. Although there are so many very important applications of those two optical manipulation techniques, there are still two requirements to generate large optical force. First, we need a high power laser beams. Secondly, the refractive index of the particle and the surrounding medium has to be quite different. Well, those two requirements further limit the further application of the optical manipulation techniques. We have to use complex optical setup, and only certain particles can be manipulated. Fortunately, researchers, including our group, developed a new type of optical manipulation techniques named optothermal manipulation. So basically, we can achieve a similar manipulation through light-generated temperature fields. So as you can see here, we simply use a laser beam to heat up a light-absorbing substrate. It can be plasmonic substrate. And then in the liquid, the micro nanoparticle can be attracted at the hot spot. It shows advantage like low power and simple optics, and it also features long range manipulation. Here are some examples. We can use optothermal manipulation platform for the nanoparticle manipulation. Especially in this work, we achieved the manipulation of metallic nanoparticles smaller than 100 nanometer. So that's very challenging for the Nobel Prize technique. And we also achieved the cell manipulation. You can see this is yeast cells. And also we can use it for the colloidal fabrication. And also we can do some molecular sensing. And on this page, I'll talk a little bit about the origin of the thermal forces. So that's the key to achieve the manipulation. There are so many different thermal forces, and today we will mainly talk about these two, and feel free to check my review article for the rest of the other forces. So how is the thermoelectric force generated? As you can see in the schematic, so still it happens in a liquid environment. It's in electrolyte solution, and there are a lot of cation and anion. So as we know, because of the electric static force, those two type of ions should stay together. And however, because we have a temperature field generated by the laser beam, the ions will diffuse from the hot end to the cold end. And those two ions, they have different thermal diffusion coefficients. So there exists a small separation, as you can see, marked by this box. And if we accumulate all those separation, we can get a thermoelectric field as marked by these pink arrows. And the electric field is pointing toward to the hot end. And another background information is that most of the colloidal particles are immersed in the liquid. They have surface charge because of the dissociation of surface function group. So if the particle has positive charge in such an electric field, the particle will move to the hot end, and that's the origin of thermoelectric force. And another very important force is the depletion force. So it's basically an osmotic force. As you can see in the schematic, there are some PEG molecule and also some DNA particle. So the researchers were trying to use those PEG molecule to push the DNA molecule to the center of the laser beam. So it relies on the depletion force. Again, there's a temperature field, and the temperature field make the large PEG molecule diffuse to the cold end. As you can see, there are more PEG molecule here. And then we have a concentration gradient of the PG molecule. And as we know, if there are a lot of solute particles in the solvent, there are larger osmotic pressure putting on the membrane. So similarly, there will be larger osmotic force putting on the DNA molecule. So that's the depletion force. So those two forces are very important in my two projects. And that's why I make a brief introduction here. And last page of the introduction part is the current limitations of optothermal manipulation techniques. So despite the promising future of the optothermal manipulations, there are still some limitations. There are actually two major limitations. So first of all, let's take a look at the magnetic robots platform developed by another researcher. So they basically achieved the pulling, thrust, and rolling of three different particles and conducted a complex surgery to the mouse. So basically, they achieved the multimodal manipulation of magnetic robots. However, for most optothermal manipulation platform and also most optical manipulation platform, they only enable one manipulation mode. It can be tweezing. It can also be pushing, like basically push the particle away the laser beam. It can also be rotating, but like there's no single platform enables more than 3 different manipulation modes. And another thing is that for most all the optothermal manipulation platform, they need custom substrates. You can see in this work, they need the gold substrate with those nano holes. And this work, they need a very ultra thin chamber in order to trap the particle in Z direction. And this is one of my works, and we need the plasmonic substrate in order to generate temperature field. So those are the two limitations. And my first project is to overcome the first limitation to achieve multimodal manipulation. And as you can see in the schematic, so basically by adjusting the distance between the particle and the center of the laser beam, we can achieve tweezing, printing, rotating, rolling, and shooting, basically 5 different manipulation modes. Yeah, let's quickly take a look at the mechanism. You can see the particle, it's immersed in a liquid environment. The liquid environment, there are PG molecule and also some sodium chloride ions that I did not plot in this figure. Under the temperature field, there are 3 different forces. The first one, it's the electric force from the substrate. So as I mentioned before, the particle have surface charge because of the dissociation of the surface function group. And for this substrate, we also deposit a thin layer of molecule called BSA. BSA has a specific property. It has a very large surface charge, and the surface charge can be tuned by the temperature. So there exists the electric interaction between the particle and the substrate. The second force is the thermoelectric force that comes from the ions, specifically the separation of the charges is from the thermoelectric force, thermoelectric field. The last force is the depletion force, which is basically the osmotic force that I introduced it before. And the tweezing or printing and rotating or rolling, shooting, those 5 different movements of the particle are actually the results of the synergy of these 3 forces. So for example, for tweezing and printing, the depletion force is dominated. The depletion force basically pushes the particle down to the substrate and at the hot spot. But for the rotating case, if we have a small distance between the particle and the center of the laser beams, we can achieve a force balance among the three forces, and there's only a net torque. That's how we achieve rotating. And if we just increase the distance for a little bit, then the force balance will be broken, and there's a small net force that push the particle away from the laser beam, and meanwhile there's still a torque. And if we further increase the distance, at this case, the torque disappears, and there's only a small net force that pushes the particle away from the hotspot. So those are the general concept of this work. Then we can take a look at some experimental results. So you can see this is a video showing how we manipulate the particle, and the cross marks the position of the laser beam. You can see we just move the particle around, and those successive images shows how we move the particle around. And then printing. So there are two particles. Then we move the laser beam to the target particle and turn on the laser beam for a few seconds. After that, we turn off the laser beam, and you can see the particle is completely still. And in contrast, the reference particle is still moving around. So basically, it proves that we successfully print the particle on the substrate. And then we achieved the rotating, rolling, and shooting. So there are some videos. This is rolling. You can see the two fluorescent small bits clearly show it's rolling. And then shooting, it's a pure translation. Yeah. So next step, we want to do some more accurate control of the particle. So my colleague, Zhihan, he helped me to establish this feedback control system. Basically, we add an additional device named the spatial light modulator. This device can help us to more accurately control the position of the laser beam through some electric signal. And he also helped me to write an awesome algorithm to help to do some real-time monitor of the position of the particle, so that we can simply use some simple words as an input to control the movement of the particle. So here are some videos. You can see we control the shape of the four particles, and basically the shape is changed from this one to diamond, and then use the shooting mode to enlarge the diamond. And moreover, we can control the movement of 200 nanometer gold nanoparticle, and basically we use it to write UT Austin. And that's pretty cool. Last piece of this project, we achieved the characterization of cellular surface. That's the most exciting part of this work. Basically, it shows some promising biological applications of our platform. So let's directly look at the video. So first, we rotate this Easter cell and then print it on the substrate. Then we bring another smaller cell, the smaller cell functioning as a probe cell. And we use rotating mode to rotate the probe cell at the surface of the target cell, basically rotated at different sites, because we want to measure the cell-cell interaction. Because the target cell has a uniform surface, because the only non-uniform area has been rotated up. So all the three different contacting sites has uniform surface property. And that's why we get almost the same rotation rate at 6 different sites. In the second video, you can see still we just repeat the experiments, but this time the target cell, there are 3 different non-uniform regions that we call bar scar. The 3 region has basically larger attraction to the probe cell. So the friction force is larger. And again, we conducted the rotation at 7 point, and those figures shows the statistic data. Basically, if the contacting point at Bhatt-Scar region, the rotation rate is smaller because of larger friction force. So it's a proof of concept application, basically show that we can use our platform to measure the surface property. Yeah, quick summary of this project. For the first time, we achieved the multimodal optical manipulation, and we also achieved the intelligent self-navigation system for the control of particle. And also it shows promising biological applications. Yeah, I think we are running a little bit late, so I will wrap up the second project quickly. So basically we achieved the multimodal manipulation on arbitrary surface. Firstly, all the experiments happens in a plasmonic substrate, so there are definitely some limitations. Yeah, the second project, as you can see here, the manipulation, still we achieve the multimodal manipulation. And by this time, we can achieve the manipulation in a glass substrate. And moreover, on the surface of some live animals. We will dive into it deeply soon. And so as you can see here, this page shows the general concept. So the difference compared to the previous project is that we established the temperature field on the particle instead of the substrate. As you can see, this is the measured temperature distribution. And again, we achieved the tweezing, rotating, rolling, and also shooting, and I do not put it here. The major difference, as I mentioned, is the surface. It can be arbitrary surface. Second difference is that we do not rely on the substrate force. Instead, we use optical force. And the different manipulation, it's due to the synergy of these three forces. Here are some images to show different manipulation modes. I will go through it quickly. Basically, those manipulations are quite similar compared to the previous one. And then this page shows the manipulation of single cell. So for this one, you can see we control this synthetic particle and pick up this yeast cell, and then bring the yeast cell to the target position. That's how we achieve the delivery of the single cell. And moreover, we can achieve the rotation of the single cell. You can see here. And more exciting thing is that if we have two cells contact with each other, by shifting the position of the laser beam, we can achieve the manipulation of single cell. You can see in the first row, we just rotate this cell and leave the right cell at its position. And in the second row, we can achieve the rotation of this cell and keep the left cell at the original position. And the last piece of this project, it's we achieved the manipulation on top of a warm embryo. It's a live warm embryo. You can see we manipulate the particle on the top of it. And after the manipulation, the worm can still hatch. So that's definitely non-invasive manipulation. And more exciting thing is that there's a live worm, and we can manipulate the silicon nanoparticle on top of it. And there's a video, so we can have a more straightforward impression of it. So we bring the silicon nanoparticle on top of it and then move it around. And meanwhile, the worm is still moving around. Yeah, last piece of it. it's some in vivo manipulation. Basically, we inject a silicon nanoparticle inside a live warm egg and then move it around. So you can see there's a small particle. Then we gradually nudge the particle and then suddenly increase the optical power to increase temperature and meanwhile increase the force so that we can open a little bit the hole on the warm egg and then inject the particle inside. So the interesting finding here is that when we illuminate the laser on the particle, the worm beat its tail at a high frequency. In comparison, if we shine a laser beam simply on the worm instead of the particle, the frequency is lower. And if we completely turn off the laser beam, the tail beats frequency is lowest. And for the study of worm, there are a lot of researchers, they are currently studying the thermodynamic study of the worm because it's related to the cell division. So our platform shows potential application for that. Yeah, I think that's the end of the presentation. I would like first thank my supervisor, Professor Yue Bingzhen, for his continuous guidance and a lot of support to my research and beyond. And also I want to appreciate my collaborators and also my group members, also the funding agency. And last but not least, I joined Intel last month and I'm happy to connect if you want to have any questions regarding my research. I'm happy to connect. And that's all about it. Xiang and Huaizhi, if you have any question, feel free to ask.
Yeah, thank you so much for your amazing presentation. The topic is really, really interesting. I wish we schedule for more time for your seminar. Yeah, we'll wait a couple seconds to see if any questions from our audience.
Yeah, I have one question.
Huaizhi, are you saying something?
Yeah, can you hear me? I got a question.
Yeah, please go ahead.
I'm curious about what's the laser intensity you use for the optothermal manipulation, and roughly like what's the temperature we will rise to, like how many degrees? And yeah, that's my question.
Yeah, for the second project, as you can see here, actually for different experiments, we have different optical power and different temperature. But roughly the temperature rise is between, I would say 5K to like 15K. So specifically for colloidal particle, we can use higher temperature, but for some cells, we would try our best to minimize the temperature rise. And the optical power, you are asking the optical power or optical intensity?
It's better if you could provide like both, like the power and then roughly the the area.
You mean the beam spot?
Yeah.
I cannot remember exact the number, but the optical power is typically let me see. The smallest one, I would say only roughly 10 microwatt, and but for the maximum power, it can be as high as 0.1 milliwatt. And for the beam size is typically ranges from 0.7 micrometer square to 3 micrometer square. Yeah, that's the numbers that I remember. And feel free to check my papers. There should be some more detailed numbers.
Okay, thank you.
Thanks for the question.
And yeah, I also have some questions. I saw that the particles in the video moves around. So I'm curious, is Brownian motion a problem in your system?
Yeah, that's a great question. So let me see how I can. So for the Brownian motion case, for example, if we are tweezing the particle, the force is basically trap the particle to the hotspot. So if we increase the optical power, we can generate a larger temperature field. It can give us a larger force that can definitely help us to suppress the Brownian motion. And another thing to reduce the Brownian motion is the PG molecule. So this molecule, it has high viscosity. So the viscosity can kind of help us to suppress the Brownian motion. And Brownian motion is very, very important for the particle manipulation. And you can see the minimum, the smallest particle that I can manipulate is roughly 100 nanometer. The reason why we cannot trap particles smaller than that is the Brownian motion. Brownian motion is so strong for smaller particle that the trapping force is no longer be able to dominate the movement of the particle. Yeah.
I see. Oh, yeah. Thank you. Thanks for the clarification. And yeah, a follow up with the previous question. Yeah, for these slides, I see that for different d, the distance, it will affect what mode you're
yes,
you affect the modes. Yeah. So I'm curious, do the particle size affect the manipulation mode as well?
Yeah. That's another great question. So we do have some study of the size effect.
Yes.
So for smaller particle, the d will be smaller. But again, I mean, we can achieve a similar multimodal manipulation on smaller particle.
Yeah.
But the difference is that the optical power will be different. Typically, for smaller particle, the power will be lower, and the d will be smaller.
Okay, so you have like a table or a way to, if you use different size of the particle, you can adjust the settings that you can exert the correct mode on it.
Yeah, we hope to have the table, but I think we only have a figure to show some to show the dependence on the size. But it will be kind of challenging to have a very comprehensive table because we don't have like too many different types of particle, and also the measurement is kind of time consuming.
Okay, yeah. My last question is, I'm curious about what is the largest particle you've been manipulated?
Yeah. So yeah, I think the largest one that I ever manipulated, it's single cell. It's basically a T cell. The size is roughly 20 micrometer in diameter.
Wow.
But for colloidal particle, the maximum particle that I can manipulate is smaller, because for colloidal particle, typically the density will be slightly larger. So the particle will automatically, due to the gravity, will have a larger gravity. So when it's close enough to the substrate, it will stick to the substrate, and we are not able to manipulate it. Yeah, to answer your question, for colloidal particle, it's less than 10 micrometer, and for single cell will be 20 micrometer.
Okay, wow, that's very cool.
Mhm.
Okay, so if you have any further questions, you can email us and we'll deliver your question to Hongru. And back to you soon. I want to thank you again for your awesome presentation, and I wish you all the best in the future work. And I would like to thank our audience as well for joining us today. Our next talk
Wonderful.
is given by Sky Price on April 20th, 2 weeks from today. We're very much looking forward to see you again. And thank you again, Hongru.
Thanks.